Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing University of Hyderabad PhD entrance examination question paper for the subject Biotechnology, and it was conducted in the year 2020. It was for PhD admission 2021 session. I will be discussing only the biology part. Then first question. Reductases are enzymes involved in. So reductases are add hydrogen to substrate. So the answer is option C. Then next one. Which one of the following is considered as the father of modern neuroscience? So it is answer is option D. Option Sir Santiago Ramon y Cajal. Then next one. If there exists an X-linked recessive disorder in man who marries a normal woman, then which of the following statement is or are true? So the man is having X-linked recessive disorder and a woman is normal. So which of the following can be true? All the male children of the couple would have no risk to the disease. Yes, that can be an option. So one is correct. 50% of the male children of the couple would have no risk to the disease. That is wrong. All female children will be deceased. No, all female children will be carriers. Answer is option C, that is 1 and 4. Then next one. Put the following compounds synthesized during citric acid cycle in the correct order. So first one will be succinate. Then comes fumarate, then comes malate, and then comes oxaloacetate. So answer will be 4, 2, 3, 1. Then next one. Match the following. Strong anion. It's quaternary amine. Then next, weak anion. Weak anion is tertiary amine. Strong cation. Strong cation is sulfonic acid. And a weak cation is carboxylic acid. So answer is 4, 3, 1, 2. Option D. DNA from organism A labeled with N14 is hybridized with an equal concentration of DNA from organism J labeled with N15 and then centrifuged to equilibrium in cesium chloride. 5% of the total renatured DNA has a hybrid density. What fraction of the base sequence is common to the two organisms? So here only 5% has been renatured and the rest 95% have not. So only very less base sequence will be common to two organisms. So that will be around 10%. That is the least value they have given in the options. So answer is option C. Then next one. Which of the following statement is not correct for components used in SDS page? So DTT is used as a reducing agent. That's correct. SDS is used as cationic detergent. No, it is an anionic detergent, not cationic. Then bromophenol blue is used as a tracking dye. That's also correct. Kumasi brilliant blue is used for protein staining. That is also correct. So the answer is option B, that is SDS is used as cationic detergent. That is the not correct statement. Then next one. The phenomenon which shows lack of correlation of genome size and the complexity of an organism. So it is C value paradox. Answer is option B. Then next one. A typical gene utilizes a segment of DNA whose molecular weight is 1 million. How many turns of the helix do this represent? So they have asked for number of turns of the helix or number of base pairs per turn. So molecular weight they have given as 1 million and one base pair is 660 grams per mole. So 1 million will be how many base pairs? That will be around 1500 base pairs. So there is 10 base pair per turn. So 1500 base pair will have 150 turns. So the answer is option B, that is 150. Then next one. Which of the following technique uses immobilization strategy? So it is answer is option C, that is surface plasmon resonance. Then a molecule has more covalent character if. So it is having more covalent character if charge on either of the ion is high. Answer is option D. Then next one. E. coli chromosome completes the entire cycle of DNA replication in 40 minutes. If the molecular weight of bacteria is 2.6 into 10 raised to 9, find the average rate expressed in nucleotides per second of the replication fork that progresses along the chromosome. So it is given by molecular weight of DNA is equal to number of base pairs into average molecular weight of nucleotide pair. So number of base pairs will be molecular weight of DNA divided by average molecular weight nucleotide pair. So 2.6 into 10 raised to 9 by average molecular weight of nucleotide pair is 660. That you can get an approximately around 4 million. Now one base pair is equal to 2 nucleotides. So this many base pairs will be into 2, so that will be 8 million. That is average rate, that is nucleotides per second. Here it is 40 minutes. Convert 40 minutes to seconds, multiply by 60 and you will get 2400 seconds. Divide 8 million by 2400, you will get this value which is approximately 3600 nucleotides per second. Answer is option C. Then next one, match the following. Affinity chromatography, that is trypsin sapphirose 4B. Anion exchange chromatography, DEAE cellulose. Cation exchange chromatography, CM cellulose. Gel filtration chromatography, Cephedex G450. Answer is option C, 4123. Here they have given the chromatography and the stationary phases that they have used in chromatography for column preparation. Then next one, which of the following recognize a specific amino acid and its cognate tRNA molecule? So it is tRNA synthetase. When you are learning protein translation, you will find this. Answer is option D. Then next one, the scientists H.J. Alter, M. Houston and C.M. Rice were awarded Nobel Prize in the year 2020 for their contribution on the following virus. So it was hepatitis C virus that they have worked on. Then next one, the pH of a solution is 4. HCl is 4. What is the molarity of the solution? So pH is equal to minus log concentration of H plus minus log concentration of H plus, that is 4. So 10 raised to minus 4 will be 0.001 molar. So answer is option C. I made a mistake over here, okay. This one 4, this 4 has to be put over under here, not in log 4. So concentration of H plus will be 10 raised to minus 4, that will be 0.001 molar. I made a mistake there, please correct it. Then next one of the following gives us measure of the irreversibility of a given process, meaning also that is a measure of disorder of a system. So it is entropy. It is the measure of disorder of a system, entropy. Then among the different cell-based expression system, which one is not free from the contamination of endotoxin? So it is bacterial cell that is not free from contamination of endotoxin. Answer is option C. Then next one. The following constitute important mechanisms of genome evolution in bacterial lineages. So first one is horizontal gene transfer, genome reduction, pseudogenization, codon usage bias. So of these, the important mechanisms of genome evolution in bacterial lineages is horizontal gene transfer, genome reduction and pseudogenization. That is 1, 2 and 3, whereas codon usage bias is not. Then next one. ZMWs of the SMRT cell. That is single molecule next generation sequencing. 1, 3. Ion chip, a specialized silicon chip. NGS by pH change monitoring. Illumina his seq in 2000. So that is cluster generation and bridge amplification and sequencing by synthesis with reversible dye terminators. 454 genome sequencer, that is pyrophosphate detection. Answer is option D, that is 3142. Next one. The hydrogen bond formed by a polypeptide chain in pi helix can be represented as. So hydrogen bonds with the pi helix display repeating pattern in which the backbone, that is C double bond O of residue I, hydrogen bonds to the backbone NH of residue I plus 5. So answer is option C, that is I giving I plus 5. In your answer key it is option D, but when I referred other books I found that it is I giving I plus 5. Then next one. Transient interaction of proteins are expected to control the majority of cellular processes and are best studied by. So they are transient interaction involve protein interaction that are formed and broken easily. So that is transient interaction of proteins, and they are best studied by cross-linking reagents. Answer is option C. Then next one. The instrument used to measure the light intensity, it is photometer. Answer is option B. Then Louis Pasteur worked on anthrax. Answer is option D. The basic unit for Baltimore system of virus classification is, it is mRNA formation. Answer is B. The factor which does not affect pKa value of an amino acid is the loss of charge in the alpha carboxyl and alpha amino groups. Yes. The interaction with other peptide R groups. Yes. Other environmental factors. Of course. So the left out is molecular weight. Answer is option D. Then the following is a cell line used in vitro experiment to serve as an experimental model for human monocytes. So that is THP1. Answer is option A. Then what best defines the genomic island occurring in bacterial chromosome? They have different G plus C composition than the chromosome as a whole. They are reminiscent of retroviruses, usually inserting in tRNA genes and are flanked by mobilizable elements. They are almost always associated with imparting a fitness advantage to bacteria. So a genomic island, it is a part of a genome that has evidence of horizontal origins. And a genomic island, it can be involved in the symbiosis or pathogenesis, and it helps the organism in its adaptation to that particular environment. So the answer here is they have different G plus C composition than the chromosome as a whole. That is correct. One is correct. They are reminiscent of retroviruses. No. Usually inserting in tRNA genes and are flanked by mobilizable elements. That is also correct. They are almost always associated with imparting a fitness advantage to bacteria. It is not any fitness advantage. They help the organism in adaptation. They are involved in symbiosis or even pathogenesis also they are involved. So they are not associated with imparting any fitness advantage to the bacteria. So the answer is option D, that is 1 and 3. Then next one. Match the viruses based on the genetic material they carry. HIV, it carries single-stranded RNA. Then reovirus, it carries double-stranded RNA. Then lambda phage, it carries single-stranded DNA. And double-stranded DNA. Lambda phage it carries double-stranded DNA and parvovirus it carries single-stranded DNA. So the answer is option C, that is 3, 4, 1 and 2. Then next one, match the following. PDB. PDB is a database of the Drosophila genome. 1. No, not a genome. It is a resource of 3D structures of biological molecules. It is 4. Okay, I made a mistake. Protein database, PDB. It is a resource of three-dimensional structure of biological molecules. Then OMIM. It is genetic information of human diseases. Then PAM. PAM is a substitution matrix and Flybase is just a database of Drosophila genome. So the answer is option B, that is 4, 3, 2, 1. Then next one. The word fulminant refers to. It refers to severe and sudden onset of disease or symptoms. So it is answer is option D, that is quick and high mortality. Then next one. You are setting up an in vitro experiment to study DNA replication at the replication fork. In each of the following tubes, you have set a series of reactions by adding the enzymes and or reagents that are required for DNA replication. However, omitted only one of the components as mentioned below. So in first one, there is no ligase being added. In second one, no primase was added. Third one, only DNTPs were added, no NTPs added. And in fourth one, no DNA polymerase was added. So indicate in which case DNA replication will be affected. So answer is option 2, that is no primase added. Yes, if primase is not added, DNA polymerase cannot start synthesizing. Then only DNTPs were added, no NTPs. If DNTP is not added, what will DNA polymerase keep on adding? Nothing. So answer will be option 3 also is correct. Then DNA polymerase was added, that is fourth one. No DNA polymerase was added, so it will not result in replication. There is no enzyme to carry out the replication. So answer is option A, that is 2, 3 and 4. Then next one. For the statistical validation of a phylogenetic tree, which of the following could be used for the resampling of a multiple sequence alignment? So it's bootstrap analysis. Answer is option A. Then what is the correct order of events during DNA replication of the lagging strand? So first you have answer 3, separation of double-stranded DNA by helicase. Then you have got RNA primer synthesized by DNA primase. Then binding of single-stranded DNA binding proteins. Then formation of new phosphodiester bond by DNA polymerase. And exonucleolytic proofreading by DNA polymerase. So the answer is option B, that is 3, 5, 2, 1 and 4. Then next one. An action potential involves Na plus moving dash the cell and K plus moving dash the cell. So Na plus moving inside of the cell. This is action potential, and K plus moving outside of the cell. So answer is option A. Then next one. Acetylcholine is a neurotransmitter that provides for communication between muscles and nerves. When there is a problem with the interaction between acetylcholine and acetylcholine receptor sites on the muscles, which condition can occur? So it is myasthenia gravis that will occur, and it is an autoimmune disease of the neuromuscular junction which results from antibodies that block or destroy acetylcholine receptors at the junction between nerve and the muscle. So answer is option A. Then next one. Dopamine is an important neurotransmitter. Which disease or disorder results when the neuron in the brain that produces dopamine die? So it is Parkinson disease that will be the result. Answer is option C. So coming to the next question. Consider the following statements. Eco R1 is from Escherichia coli recognizes GAA-TTC. So they have asked for which of the following statement is correct. So option 1 is naturally correct. Then you have got in a dideoxy chain termination sequencing reaction only one primer is used. So that is also correct. The northern blotting technique is used for DNA transfer and detection. That is not correct. It is for RNA transfer and detection. In DNA microarray analysis, probe is labeled with fluorescent dye. That is also not correct. So which of the following statements is or are correct? That is 1 and 2 are correct. Option A is correct. Then next one. Addition of an inhibitor to an enzyme substrate reaction following Michaelis-Menten mechanism increases the Km value but does not affect the Vmax value. Then the inhibitor could be. So they have added an inhibitor to the enzyme substrate reaction following Michaelis-Menten kinetics, and they found that there is an increase in Km value but there is no change in the Vmax value. So which inhibitor is it? So competitive inhibitor, because it increases the Km value but does not change the Vmax value. So it is the case of a competitive inhibitor. Now this is a similar question from competitive inhibitor is asked for majority of the entrance exams. So please learn all these inhibitors, competitive, uncompetitive, non-competitive, everything including the graphs for these competitive inhibitors and uncompetitive inhibitor and non-competitive inhibitor also. There even at least one single question is being found from that section. Then next one. The enzyme actively involved in scavenging of reactive oxygen species is. So alternative oxidase, yes. It is there, and in case of plant I think alternative oxidase, and then you have got superoxide dismutase. So 1 and 2 are correct. Answer is option D. Then Down syndrome results from. So Down syndrome, the presence of an extra chromosome in position 21. Answer is option B. Then which of the following statement is not correct about the NMR? It stands for nuclear magnetic resonance. That is correct. Protein samples are labeled with radioisotopes such as C13 and 15. That is also correct. Then NMR spectroscopy detects spinning patterns of atomic nuclei in a magnetic field. That is also correct. Then NMR spectroscopy detects spinning pattern of atomic nuclei in an electric field. That is not correct. So the answer is option D. Then which of the following pre-zygotic reproductive isolation mechanisms best explains why salamanders that live in trees do not successfully mate with salamanders that live in soil by rivers? Answer is option C, ecological isolation. Then next one, in DNA sequencing by Sanger dideoxy method, the specific enzymes are used to cut the newly synthesized DNA into small pieces which are then separated by electrophoresis. No restriction of DNA. So answer A is not correct. The dideoxynucleotides must be present at high levels to obtain long stretches of DNA sequence. If you have higher concentration of DDNTP, then in the reaction shorter products will be obtained that will be very much closer to the primer sequence. So with lower concentration of DDNTP, chain termination is less likely and you will get longer products. So it should be an optimum concentration of dideoxynucleotides that you have to use. So B is also not correct. Then the role of dideoxy-CTP is to occasionally terminate enzymatic synthesis of DNA where Gs occur in the template strand. That is also correct. Then the template DNA strand is radioactive. No, the dideoxy version of all 4 nucleotides will be labeled with different colors of the dye. So D is also not correct. So answer is option C. Then next one. Consider the following steps involved in PSI blast and arrange them in correct order. So the order here is, first one will be blast P search using a scoring matrix such as Blossom 62 to perform pairwise alignments of the query sequence against the database. Then construction of a multiple sequence alignment from blast P search and creation of a profile. Then the PSSM is used as a query to search database. Then next you have estimation of the statistical significance of the database matches, and then fourth one, continuation of search process iteratively, typically about 5 times. So the correct arrangement order is 2, 3, 2, 3, 1, 5 and 4. Option A is correct. Then which statement is not true of sterols? Sterols are commonly found in bacterial membrane. Sterols are important components of eukaryotic membranes and very rarely found in bacterial membrane. So that itself is not correct. Answer A. Then the following could be the cause of etiological factors responsible for the development and or progression of type 1 diabetes. So genetic susceptibility and environmental triggers, that is true. 1. Allergy to wheat gluten, no. Certain antigen causing autoimmunity, yes. 3. Lactose intolerance, no. So 1 and 3 are correct. Answer is option C. That was a very easy question. Okay. One of the significant objectives or outcomes of system biology could be. So system biology, it is the computational and mathematical analysis and modeling of complex biological system. So one of the significant objectives or outcomes of system biology is data-driven testable models relevant in discovery. Answer is option C. Then if you perform dot blot analysis of DNA sequence with itself having direct repeats, it will produce. So this is the main diagonal line. So if it is parallel to the diagonal line, then it is a direct repeat. If it is like this, then it is an inverted repeat. So the lines parallel to the diagonal line will show direct repeats. They have asked for direct repeats. So answer is option D. Then in cymographic assay aim to identify the protease inhibitors, the following statement is correct. So cymographic is an electrophoretic technique for the detection of hydrolytic enzymes, measuring proteolytic activity, etc. So an example for zygomorphic assay is protein X is embedded in page, will be digested by active enzyme Y, run through the gel. After Kumasi staining, area of degradation are visible as clear bands against a darkly stained background. So here a protein X is embedded in the page, and it will be digested by active enzyme Y, run through the gel. After Kumasi staining, area of degradation are visible as clear bands against a darkly stained background. So protease is mixed with components of polyacrylamide gel before page. That is option A. That is not correct. Then gelatin is mixed with components of polyacrylamide gel before page. No. Yes, the gelatin is mixed with components of the polyacrylamide gel before page. Yes, that is correct. Okay, but it is incomplete. There is another option with that. Okay. C. Then protease is mixed with the components of polyacrylamide gel before page, followed by incubation of the gel with gelatin. Then you won't get any result. And so option C is also not correct. We'll see option D. Gelatin is mixed with components of the polyacrylamide gel before page, followed by the incubation of the gel with a protease. So that is correct. Answer is option D. Then multiple sclerosis involved breakdown of myelin sheath. Answer is option B. Now, questions regarding sclerosis are also common in various entrance exams. So, study that also. Then Hardy-Weinberg equilibrium, that is 100% they will ask something on Hardy-Weinberg equilibrium. Most of the question papers, Hardy-Weinberg equilibrium, at least one question I can see. Okay, a 30-year-old man is a heterozygote for disease that is prevalent in the population. If the population is in Hardy-Weinberg equilibrium, then P square plus 2PQ plus Q square is equal to 1 and P plus Q is equal to 1. Which of the following is the equation that indicates the prevalence of heterozygotes? So it will be 2PQ that will indicate the prevalence of heterozygotes. Then, non-steroidal anti-inflammatory drugs like aspirin and ibuprofen act by blocking production of. So, they block the production of prostaglandins. Answer is option A. Then, dash is the sensory relay station where all sensory information except for smell goes before being sent to other areas of the brain for further processing. So, it is thalamus. Answer is option D. Then which of the following methods can be used to interpret protein-protein interaction? FASTA, no. Domain fusion method, that is Rosetta Stone, yes, that can be used. Gene neighborhood method, that is also used. Chow-Fasman method, not used. So answer is 2 and 3, option D. Then match the following. XPSI. XPSI is bioinformatics resource portal. 1, 2. Then you have got Blossom, it is a substitution matrix, that is 4. And string resource of protein-protein interaction 1. And Entris is information retrieval tool of NCBI. So answer is option B. Then which of the following is a genetic disease that causes neurons in the brain to waste away and die? So it is Huntington's disease. Answer is option D. Then next one. The following statements are correct with the reference to jasmonic acid. So, it is an essential drug, essential during pathogen resistance. Yes, that's correct. It is synthesized from arachidonic acid. No, it is from linolenic acid. So, 2 is wrong. Ricin, a bacterial toxin. Ricin is a eukaryotic toxin, not a bacterial toxin. So, and fourth one, jasmonyl isoleucine is the active hormone in the jasmonate signaling pathway. That is also correct. So, 1 and 4 are correct. Option C is correct. Then next one. Identify the correct sequence in the passage of electrons to the alternative oxidase pathway of mitochondrial electron transport chain. So it is complex 1 to ubiquinone. Answer is option A. Then match the following group specific ligands with their compounds with affinity commonly used in affinity chromatography. So avidin is biotin containing enzymes 1, 2, protein A and G immunoglobulins, phenylboronate. Phenylboronate is glycoproteins, lysine is for rRNA. So answer here is 2, 1 and 4 and 3. So answer is option C. Next one. The amino acid cysteine is synthesized from a precursor of glycolytic pathway in the following sequence of reaction. So it is 3-phosphoglycerate serine O acetylserine. Answer is option A. Then next one. The specific carotenoids implicated in non-histochemical quenching of photosynthesis. Answer is zeaxanthin and lutein. Answer is option B. So that's it. Thank you for watching my channel. Please do like, share and subscribe. Thank you.